So let's take a look at blood types. Blood types are also known as blood groups, and you've probably heard of the different types of blood types, right? A, B, AB, O, and then we combine those with positive and negative, right? What makes these different blood types is the presence or absence of stuff on the outside of red blood cells, right? So let's take a look at what we're talking about exactly. So on the outside of our red blood cells, there is stuff, right? So this is supposed to be a cell membrane here, like a red blood cell membrane, okay? Mostly made of phospholipids, but there's proteins embedded in the membrane. This is the outside of the red blood cell. This is inside the red blood cell. And you can see on the outside of the red blood cell, there's proteins and carbohydrates and protein-carbohydrate combinations. And this is very common on the outside of our cells, right? A lot of these things are important for cell recognition, right? So red blood cells have stuff on the outside of their cell membranes. We're going to call this stuff antigens, okay? My antigens are probably different from your antigens, okay? So if we mixed my blood type into yours, your defensive components might actually attack my red blood cells because they're going to think that my stuff, my antigens, are foreign invaders and they're going to attack it, which they kind of rightfully should, right? So let's say this is my red blood cell and this is your defensive component. This is an antibody, right? It's going to attack and kill my red blood cells, right? So what are antigens really, right? So antigens are proteins or carbohydrates or protein-carbohydrate combinations on the outside of a foreign cell, right? So this is maybe on the outside of a bacterium, but it could be on the outside of a donor cell, for example, okay? And the function of the antigen is that it allows our immune system to recognize foreign material and get rid of it, okay? So antigens are found on the outside of bad guys like bacteria, but they're also found on the outside of not so bad guys, like let's say you needed to get a liver transplant. And the new liver that comes in will have cells that aren't your cells. And your immune system might recognize that as foreign. It'll recognize those antigens on the outside of the liver cells, and it'll try to get rid of that liver. You don't want that to happen in that case, right? So sometimes antigens are good because it helps us to get rid of pathogens from our body, but sometimes it's bad when we're trying to do something like a blood transfusion or an organ transplant. So we have something called self-antigens. So self-antigens are the proteins and carbohydrates on the outside of our own cells, and it's how we recognize self. So our immune system will scan the outside of our cells and say, okay, I know this is a self cell because it has the protein, carbohydrate, in other words, antigen combination that I know is self. And your immune system is very specially taught to leave self antigens alone. So your immune system cells are taught to recognize self antigens and then leave them alone, right? These self-antigens are the parts that are going to be problematic, like I said, if you were to do like an organ transplant or something. So the other person's immune system will recognize those antigens as not self and will attack it. So that's why one of the things that they do during an organ transplant is immunosuppression. They basically turn off the immune system so that the organ cannot be rejected. So what do antigens have to do with blood types, right? So red blood cells contain lots of antigens on their surface, right? But the only ones that are of clinical significance, the only ones that are medically important are antigens A, B, and then something called RH, which is also known as antigen D. Turns out that they named the antigens on the outside of the red blood cells alphabetically. So A was the first antigen that was recognized on the outside of a red blood cell. Then next came B, and then there was C, and then there was D, and so on and so forth. But like I said, the only ones we care about are A, B, and D, but we never really refer to antigen D as D. We call it RH. The RH comes from rhesus monkey, and it turns out that's the animal where they discovered that particular antigen first in. Anyways, the antigens A, B, and RH are the only ones that have clinical importance. The other ones don't have any kind of, clinical significance. Oddly enough, antigens A, B, and RH don't have any physiological functions either. So you can have A, B, and RH and function fine. And you can have none of those. You can have no A, no B, and no RH and also be completely fine. So they have no real true function in the body. But they are problematic when it comes to blood transfusions, which is why we need to talk about them. Anyways, the A and B antigens are what we call glycoproteins, which are carbohydrate protein combinations. And they are very ubiquitous in nature. And what that means is that pretty much any living thing, doesn't even have to be an animal, it could be a banana, it could be a bacterium. Any living thing will have antigen A or antigen B on its surface, right? And this will become significant later. Anyways, so this is where the blood types A, B, and O come into play. Blood type A only has antigen A. Blood type B only has antigen B. Blood type AB has both antigens A and B, and blood type O lacks both of them. And then we have the RH antigen. So the RH antigen is not a glycoprotein. It's actually a large intramembrane protein. And it's not ubiquitous in nature. It is absolutely only found on red blood cells. So it's not very common except for on red blood cells. And again, this will become significant later. The presence of RH is more common than the absence. So what that means is that more people on the planet are RH positive than they are RH negative. And that's how we use the terms positive or negative. Positive means you have the RH antigen, and negative means you do not have the RH antigen. So blood types are actually named by the presence of the antigen on the surface of the red blood cell. So what I'd like you to do here is to pause the video and complete this table, right? So I've given you on the one side the different antigen combinations that are present on the red blood cells for these individuals, and you have to tell me what the blood type is. So I'll do the first one, or I did the first one for you, right? So I said this person has antigen A and antigen RH. So because they have antigen A, that means they're blood type A, and because they do have the RH, they are going to be positive. So this is someone with blood type A positive. So pause the video and go ahead and complete the rest of the table. How'd you do? Good? All right. So here's a more pictorial form of what we've been talking about, right? So if this is representing antigen A, you can see this person has blood type A. This is antigen B. This person has blood type B. This person has blood type AB because both the A and B antigens are present. And this person lacks both B and A, so that would be type O. This particular picture doesn't take RH into consideration. So now I want to talk about antibodies, because antibodies play a role with blood typing and transfusion also. So antibodies are proteins that are defensive. We've talked about them in other videos. So antibodies, what we didn't maybe talk about is the fact that they can bind to antigens, right? So antibodies bind to a specific antigen, and then they cause the cell with the antigen to go through a process called agglutination. So agglutination, you hear the word glue and agglutination? So what antibodies are doing is they cause the cells with the antigen to glue together into piles. So we can kind of see that in the picture here. So let's pretend this little bean-shaped thing is a bacterium, and the things on the surface here are the antigens, and this yellow Y-shaped structure is our antibody. So here we can see the antibody is bound to the antigen, right? And notice how through that binding process, we're causing all these bacteria to kind of like clump up together, right? And this is really important for our immune system. This process of agglutination causes things like bacteria to pile up into big piles, and that makes it a lot easier for your immune system to get rid of that pathogen from the body if all of the bad guys are piled up into one spot. So antibodies are defensive proteins that you make against very specific non-self antigens. So what that means is that you can't make antibodies against your self antigens. And this makes sense because antibodies basically kill stuff, right? They're going to help you to get rid of this bacteria. So you don't want to kill yourself. So you don't make antibodies against your own self antigens. You make antibodies against foreign antigens, right? So what antibodies do you make when it comes to the blood types? If you are blood type B, you make antibodies against the A antigen, but not B, right? You're blood type B, so you have B antigens. You can't make antibodies against B because then you'll die because these antibodies will cause all your red blood cells to agglutinate, and that's bad, okay? But you don't have the A antigen. If you're blood type B, you don't have the A antigen, so you will make antibodies against the A antigen. All right, so what I want you to do right now is to tell me what kind of antibodies will be present in the blood plasma of individuals of certain blood types, right? So to go back, right, we were just looking at the case where someone was type B blood, and so the antibody that they have in their plasma would be type A. So basically, you're going to have antibodies opposite to your blood type, okay? So I did this first one for you. Let's take a look at that. If someone is A positive, what kind of antibodies will they have? Right? Well, they're not going to have any A antibodies because if they had A antibodies, the A antibodies would bind to the A antigen, killing their red blood cells. Okay. They are going to have B antibodies, right? Because they don't have the self antigen B, so they will make B antibodies. And they are Rh positive, right? So they're not going to make Rh antibodies because if they did make Rh antibodies, they would be attacking themselves, right? So I want you to go through the remaining blood types here and determine which antibodies are present, and then you can check your work on the next slide. Don't forget to pause the video. All right, so hopefully you did good. I don't know why the antibody keeps moving. Something I want to mention is that the term antibody is often abbreviated as just the prefix anti or as big A little b, right? So like if you're talking about antibody A, they might say anti-A or ABA, which can be kind of confusing, but I wanted to mention it, right? So this top part of the picture we saw previously, and the bottom part we've seen previously, right? We knew if you have blood type A, then that means you have antigen A. But what this is showing you is the type of antibodies that are also going to be present in that person's blood plasma. So notice the AB blood group, right? If you have both the A and B antigens, you're not going to have A antibodies or B antibodies because then you'd be attacking yourself. But if you're blood type O, you'll have both types of antibodies, right? So why do we care about this whole antibody stuff when it comes to blood types, right? One of the reasons is because antibodies can actually be made in laboratories and then can be used in the process to figure out someone's blood type. So basically we use antibodies as biomolecular tools, okay? So we use antibodies on blood typing tests, right? Antibodies will bind to antigens on the surface of the red blood cells and cause agglutination. We can actually see that on a blood typing card. That's what we see here in this picture, right? So see how this blood looks very different than this one, right? So what we've done here is we've taken one drop of blood and placed it on the anti-A antibody. From the same person, take a second drop of blood and put it on the anti-B antibody, and then take a third drop of blood and put it on the anti-D antibody, right? So this is what a negative result looks like in a blood typing test. So this just looks like some dried blood. That means nothing happened, which means there's no agglutination. So that means this antibody B had nothing to bind to, meaning there is no B antigen present. But in both cases here and here, we can see agglutination has occurred, that clumping has occurred, right? The red blood cells are all clumped up because the antibody did have something to bind to. So that means the antigen is present, right? So the antibody A is bound to antigen A, causing agglutination. The anti-RH antibody did bind to the Rh antigen, causing agglutination. So this person would have blood type A positive. So let's look at that standard test for blood typing in a little bit more detail. So what you're doing is you're testing for the presence of antigens A, B, and Rh, and it requires the mixing of the blood sample on the card with some dried lab-made antibodies. So basically you have this little card, and it almost looks like a business card, and it has these little circles on it, and there's dried antibody on the little circles, right? So you usually have 4 little circle areas. One is a control with no antibody, and then you have antibody A, antibody B, and antibody Rh, right? So you put your drop of blood on each of the 4 little spots and mix it up and wait to see if agglutination occurs. So when an antibody is mixed with this antigen, what happens to the red blood cells? They agglutinate. So let's say this is our blood typing card. This is our control spot. This little splotch is antibody A. This little splotch is antibody B. This little splotch is antibody Rh. Okay. So now we're going to add a drop of blood to each of those, mix it, let it sit, and then see what happens. Okay. So based on this, what blood type do we have? So this is our control. So this is telling us what nothing looks like, right? This is what happens when there's no antibody present. So we know this is what a negative result looks like, right? So anything over here that looks like this one means it's a negative result. So obviously this one and this look very similar, no agglutination, right? But here we see clumping and here we see clumping, right? So we're positive for antigen A and positive for antigen Rh. So this person would be A positive. All right, pause the slide here, answer these review questions. When you're ready, press play and check your answers.